So, glucosamine is present mean NH2 is present. NH2 is present mean nitrogen is present. Agree?
Yes, sir.
So, chitin contains carbon, hydrogen, oxygen, and whom?
Nitrogen.
Nitrogen in the structure. Clear?
Clear, sir.
Others don't have. Others have only carbon, hydrogen, and oxygen as usual. But this is special, right? The distribution. Cellulose is a structural polysaccharide making cell wall. Whose cell wall are made of cellulose pointed to you?
Cell
Whose
walls
cell
of
wall?
plants.
Plant cell wall and the cell wall of
Algae.
algae. Under unit number 3, you will study full detail about algae. Now, just a summary. Second is starch. It's a storage polysaccharide. Cellulose is a structural polysaccharide. Starch is a
Storage polysaccharide.
storage polysaccharide. In plants, in green alga, chlorophyta, you can see starch as a storage polysaccharide. Third, it is glycogen. It is also a storage polysaccharide. In whom it is storage?
In animals.
In animals
Fungi.
and fungi.
And
Right. Pectin is making the middle lamella. It is a layer sticking to adjacent cell walls. A layer that stick to adjacent cell walls in the plant is hemicellulose. Remember, hemicellulose can be noticed only in plants. Keep that point with you. Hemicellulose, no need in full writing. You can just mark a point. Only noticed in plants. Now, pectin can be noticed in some algae also. But hemicellulose can be noticed only in plants. Okay.
Okay, sir.
Right. And finally, inulin. Inulin, if you consider, it is a storage polysaccharide. But speciality is, it is a polymer of fructose. Not a polymer of glucose. It is a polymer of fructose. There is a beautiful plant. Its flowers are very beautiful, right? And that plant is dahlia. Dahlia plant. Flowers are very Nice. It's root tubers. Root tubers. Like manioc, root tubers. They have root tubers. And those root tubers store inulin. It's a storage polysaccharide. Inulin is a what kind of polysaccharide? What kind of polysaccharide?
Storage polysaccharide.
The storage polysaccharide. So if you take these, you can clearly divide them into two: structural polysaccharide, storage polysaccharide. Cellulose, structural polysaccharide. Starch, storage. Glycogen, storage. Pectin, structural. Hemicellulose, structural. Chitin, structural. Inulin, storage. Clear?
Yes.
Clear, sir.
Good. So further, now here they are dividing polysaccharides into two, linear and branch, that I already told you, correct? Linear polysaccharides and branched polysaccharides, didn't I give you?
You gave, sir.
I gave. Linear polysaccharides have only 1,4-glycosidic bond. Branch polysaccharides have 1,4-glycosidic bond and 1,6-glycosidic bond. Have you, I think it has been separately marked here. If you like, you can mark it here. Linear polysaccharides have which glycosidic bond?
1 to 4 glycoside.
1 to 4 glycosidic bond. Branch polysaccharides have
1 to 4 and 1 to 6 glycoside. bond.
for branch polysaccharides include hemicellulose as one more example. Okay. For branched polysaccharide include hemicellulose as an additional example. That is syllabus. Right. Now we come to a small briefing regarding cellulose. Cellulose is a linear polysaccharide. If it is a linear polysaccharide, what kind of glycosidic bond should be there?
1 to 4 glycoside.
1 to 4 glycosidic bond should be there. Between one molecule and another molecule, there will be hydrogen bond. Between one molecule and another molecule. Now this is one molecular chain. This is another molecular chain. This is another molecular chain. Between these chains, there will be hydrogen bond. Starch, as you are already pointed, there are two types of molecules present in the starch. One it is amylose, other one is amylopectin. Amylose is linear, amylopectin is branched. Branching places has 1,6-glycosidic bond. So this is 1,6-glycosidic bond. Label it. This is 1,6-glycosidic bond. This is 1,4-glycosidic bond. This place you label it as 1,6-glycosidic bond. Okay.
Okay, sir.
So this is glycogen. Once again, here we have 1,6-glycosidic bond. These ones are 1,4-glycosidic bond. Okay. In the functional polysaccharides, function-wise, polysaccharides are divided to two types. One we call structural polysaccharide, other one, cut this word and write storage polysaccharide. Functional, yeah, it's functional, but what is the function? Storage is the function. So it is a storage polysaccharide. Right. Who are the ones categorized here as structural polysaccharides? Mean already known ones are there. Cellulose, hemicellulose, pectin, chitin, those ones are there. But newly one is mentioned to you. What is that new one? This one. What is it? What is that chemical?
Peptidoglycan.
Peptidoglycan. Put a hyphen and write down. Polymer of glucosamine. It's another polymer of whom?
Glucosamine.
Glucosamine. So glucosamine's polymer mean it should have nitrogen in its structure. Understood?
Yes, sir.
Understood? So nitrogen containing polysaccharides, two are in the syllabus. What are the two? Nitrogen containing polysaccharides in the syllabus?
Glucosamine and peptidoglycan.
Glucosamine and peptidoglycan. Clear?
Yes, sir.
Right. And storage, that is already a known one. A small comparison between monosaccharides, disaccharides, and what's it? Polysaccharides. Among these, who don't have glycosidic bond?
Monosaccharides.
Monosaccharides, okay, they don't have glycosidic bond. Remember, glycosidic bond can be broken by carbohydrates enzymes. Who can break the glycosidic bond?
Carbohydrate enzymes.
Carbohydrates. Who can break the glycosidic bond?
Carbohydrates.
Carbohydrates. So that is what we call by the name digestion. Certain of these, now for an example, cellulose cannot be digested within human body, but certain fungus, they secrete cellulase enzyme and digest it. Understood?
Yes, sir.
Inulin cannot be digested in human body, but certain bacteria secrete inulase enzyme and digest it. Like that, all these components can be digested except monosaccharides. Monosaccharides cannot be digested. Reason, they don't have glycosidic bond. So there's nothing to break them. Clear?
Yes, sir.
Right? These kind of points will be useful for you in the MCQ papers. Okay? In the A level, the most challenging and the most beneficial area is which one? MCQ. You will be getting 50 MCQ. Out of 100, you will score 50 marks in 50 MCQ. So 50 marks in 50 MCQ mean every MCQ is worthy, right? Okay, for MCQ, small points are very, very important. Those kuti points, okay, we have to keep in our memory. We have to practice it. Then only, okay, MCQ paper will be well worked. If it is well working only, your score will be topping. Here, we have to understand one thing. Now, yesterday, not yesterday, day before yesterday, A-level results was released. One of my guy in Kandy district, he has got 3A. The point it is, taking 3A, he is in the 109th rank in the district. Understood? Taking 3A, he is in which rank? Which rank?
109.
109th rank. Generally, about 100 people only getting medical faculty. So he is in the border, okay, with suspicion whether he will get the medicine or will not get the medicine. In that confusion, he is there. See, after taking three A's, that child is not happy. Understood? Understood?
Yeah.
Yes, sir.
So you have to understand the severe competition in front of you. that severe competition should not allow you to, okay, sleep. Then only you are on the track. This much competition there, this many people are fighting for it. I have to place myself within 100. How can I enjoy everything around me? If that attitude is developing, there your success starts. Understood?
Yes, sir.
That is the place your success starts. Okay? What comes in the dream, what comes in the sleep is not dream. The one that doesn't allow you to sleep is called dream. Your dream should not allow you to, what is it? Sleep calmly. All the time it should alarm you. I have a competition. I have to win this competition. There are so many people competing. Nearly 10,000 people competing. I have to place me within 100. How can I stay calm? Okay? That attitude you have to build inside your body. In your body, you have to build that attitude. Then gradually you will get adapt to it. Okay. So among these, who is not digestible?
Monosaccharide
Monosaccharides, because they don't have glycosidic
bonds.
bond. Water solubility, monosaccharides and disaccharides are water soluble. Polysaccharides are not water soluble. Monosaccharides and disaccharides are sweet in taste. Polysaccharides, they don't have any taste. They can be, monosaccharides and disaccharides can be crystallized, crystallization. Polysaccharides cannot be crystallized, right? Monosaccharides and disaccharides, if you consider, all monosaccharides are reducing sugars. Disaccharides, except sucrose, all the others are reducing sugars. Polysaccharides are not reducing. Okay. Sucrose is not reducing. Some children ask, sir, why sucrose is not reducing? Actually, it's an unnecessary question, but as a teacher, I should not tell the students it is an unnecessary question. Actually, in a reducing sugar, there will be an alcohol region that can undergo oxidation. If that alcohol region is available only, it can undergo oxidation, then it can work as a reducing sugar. In sucrose, those alcohol regions are utilized to make the glycosidic bond. Because they are utilized to make the glycosidic bond, those alcohol regions are not available to undergo oxidation. Because they cannot undergo oxidation, they cannot work as reducing sugars. So this is the fun idea, but nothing is important there. No one is going to ask you why sucrose is a non-reducing sugar? Are you clear?
Yes, sir.
So if you get to know, sucrose is the one and only non-reducing sugar. That is enough until you finish your A levels. Right. Peptidoglycan is a special polymer. It has glucose amine. In its structure, some amino acids also can be seen. Between the glucose amine molecules, what kind of bonds will be there? Between the glucose amine molecules, what kind of bonds will be there?
Glycosidic bond.
Glycosidic bond will be there. There are some amino acids also in peptidoglycan. If amino acids are present, between the amino acids, what kind of bonds will be there? Peptide bonds will be there. Clear?
Yes.
Right. And finally, glycosidic bond. This is the definition for glycosidic bond. This is the definition for glycosidic bond. In your definition book, you need to update it. Understood? Good. So with this one, carbohydrates are finishing. And we are starting. What is it? Lipids. In the case of lipids are a heterogeneous group of compounds. A heterogeneous group of compounds. Okay, they are, if you consider lipids have two major properties. One, it is, I think it's already written, but one, it is, they are hydrophobic. Okay, here you write down characteristics of lipids. One, hydrophobic molecules. They hate water. They don't mix with water. Okay. Second, the second one, they are large biological molecules. How many properties? How many properties?
Two.
So, third one, they are not macromolecules. You know macromolecules, right? Biopolymers. They are not polymers. They are not polymers. So they are hydrophobic molecules. They are large biological molecules. They are not macromolecules or biopolymers. Have you marked it?
Yes, sir.
Yes, sir.
Good. Here, you are pointed a set of categories of lipids. In the categories, okay, 3 are the major ones in syllabus. That means in exam paper, you have to give them the priority. One, it is, you have to write triglycerides or fats. What is the first category? What is the first
Triglycerides
category?
or fat.
Triglycerides or fats. Second one, whom?
Phospholipid.
Phospholipid. Third one, whom? Whom?
Steroid.
Steroids. These 3 are the ones primarily mentioned in the syllabus. Another 2 are there. They are also lipids. You should know them as lipids. But you should not give priority to write them as answers. Sometimes in an MCQ paper or anything, if those words are coming, you should be able to understand and apply them. Clear?
Clear,
So
sir.
in exam paper, say they are asking what are the major 3 categories of lipids. What will you answer? What
Fats,
are the
sir.
3 mean? What will you answer?
Triglycerides, phospholipids, steroid.
Even if you write fats, phospholipids, steroids, okay. Understood?
Yes, sir.
Fats, phospholipids, steroids, if you write, perfect. No problem. Marks will come. Right. So if you take lipids, in the lipids, carbon, hydrogen, oxygen are the primary elements. In the phospholipids, nitrogen, phosphorus like elements can be noticed. Phospholipid mean surely it should have phosphorus, right? That's why it is called phospholipid. Am I right?
Yes, sir.
Yes.
In addition to phosphorus, there will be another component, nitrogen also can be there. If you take hydrogen to oxygen ratio in the lipids, it is very high. That means oxygen's number will be very low. Hydrogen's number will be very high. So between hydrogen and oxygen, if you develop a ratio, it will be a bigger number, something like 18:1, like 35:1, like bigger ratios, and no exact ratio. 2:1 in carbohydrates is a fixed ratio, right? Standard ratio. That kind of a standard ratio we cannot notice in the lipids. Okay. The second point is telling you they are hydrophobic. They hate water. And they are not polymers. Remember, although they are written as the third and fourth point, they are the main points. That's why I gave you separately. What are the characteristics of lipids mean? What do you have to write? They are hydrophobic. They are large biological molecules. They are not macromolecules. Those are the ones you have to bring forward. Clear?
Yes, sir.
Good.
Yes, sir.
Good. And their density is less than water, so they float on water. They don't have color. They don't have odor. and they don't have any taste. Okay, so there are very few points to you regarding lipids. Fine. Now we are coming to the next heading. What is the heading to you? What is the heading to you?
Triglycerides.
Triglycerides. Triglycerides have another name also. Mark that name. Triacyl glycerol. Triglycerides have another name called, what is it? Triacyl glycerol. Okay,
Okay.
right.
Okay, sir.
In making triacyl glycerol, we will hereafter study that one in full detail. Okay, it is the major group, and fat and oil are coming to that category. 3 fatty acids and 1 glycerol molecule take part in making this molecule. 3 fatty acids and 1 glycerol molecule take part in making them. Here, there will be ester bond formation. What is the characteristic bond in lipids? What is the characteristic bond in lipids?
Ester bond.
Ester bond. What is the characteristic bond in carbohydrates?
Glycosidic bond.
Glycosidic bond. Okay, right. Lipids, if they are found in solid form in room temperature, they are called fats. Lipids, if they are found in the liquid form, they are called, what is it?
Oils.
Oils. Right. And they can be digested by which enzyme?
Lipase.
Lipase enzyme. Okay, right. Now it is time to introduce one by one. First I am introducing whose structure? Whose structure?
Glycerol.
Glycerol. It is this molecule. This group is called hydroxyl group. This is a hydroxyl group. This is a hydroxyl group. This is a hydroxyl group. How many hydroxyl groups are present in a glycerol molecule?
3.
3 hydroxyl groups are there. 3 hydroxyl groups are present mean 3 ester bonds it will form. How many ester bonds?
3 ester
How
bond.
many ester bonds it will form? 3 ester bonds it will form. So mark the hydroxyl group. You can just commonly label all 3 together as which group, or we can put a circle around one OH and say it is hydroxyl group. Okay, have you marked it?
Yeah, sir.
Yes,
Good.
sir.
Now, my children, we are coming to the home. fatty acid. Whom it is? Fatty acid. If you take a fatty acid molecule will be a molecule like this. Okay. Ayyo. Right. I will take this. This is fine. Here, look at this molecule. Don't see the down one. Down one is not your limit. Okay. If you take this molecule, this is a carboxyl group. What kind of group?
Carboxyl group.
Carboxyl group. It's a acid group. It's a polar group. Same time, this rest is a hydrocarbon chain. So what are the parts making a fatty acid?
Carboxyl group and
A carboxyl group and a hydrocarbon chain. Could you please take a white sheet, mark this diagram and label it? Carboxyl group, polar group, hydrocarbon chain, non-polar. Could you please do it? Tell me after completing. Completed?
Yes.
Yes, sir.
Right. Now, Now remember, as I told you, the carboxyl group is a polar part or non-polar part?
Polar.
Polar part. But hydrocarbon chain is a
Non-polar.
non-polar part. But the majority
Non-polar.
the non-polar. So overall fatty acid will be a non-polar component. In the hydrocarbon chain, look at this one. In this, okay, in this molecule, this is the carboxyl group. Okay. Then the rest is going to be the
Hydrocarbon chain.
hydrocarbon chain. Correct? In the hydrocarbon chain, between carbon atoms, at least in one place, if there is a double bond, then it is coming under what kind of fatty acid?
Unsaturated fatty acid.
Unsaturated fatty acid. If all the carbon bonds in the hydrocarbon chain are single bonds, then it is coming under
Saturated fatty acid.
saturated fatty acid. Clear?
Yes, sir.
Good. So, in a lipid, if saturated fatty acids are making the majority, then that lipid also will be called saturated lipid or saturated fat. If majority of the fatty acids are unsaturated fatty acids, then how will you describe that fat? How will you describe that fat? Tell me.
Unsaturated fat.
Unsaturated fat. But definition wise is easy. What is saturated fat? Fats that are made up of saturated fatty acid. What is unsaturated fat? Fats that are made up of
Unsaturated fatty acid.
unsaturated fatty acid. What are saturated fatty acids? They are fatty acids with hydrocarbons chain or hydrocarbons having no any double bond. What are saturated? Okay, having no any double bond. What are unsaturated fatty acids? They are fatty acids having hydrocarbons having one or more. What is it?
Double bonds.
Double bond. Okay, saturated fats are in which physical state in the room temperature? Saturated fats are solids in room temperature. Unsaturated fats are majorly in which physical state
Liquid.
in the room temperature? They are
Liquid.
liquids in the room temperature. Understood?
Yes,
Are
sir.
you
Yes,
clear?
sir.
Are you clear?
Clear, sir.
Right. Now, my children, we are coming to cis unsaturated fatty acids and trans unsaturated fatty acids. What are the two types of fatty acids? What are the two
Cis
types of fatty
and
acids?
trans.
The unsaturated fatty acids are two types. Unsaturated fatty acids are two types. What are the two types of unsaturated fatty acids? Easy word. One we call cis, other one we call
Trans.
trans. Look at this group. If this group is there, majorly these two hydrogen atoms should be in one side, then it is called cis or trans.
Cis.
Cis. If one hydrogen is in this way, another hydrogen is this way, like a cross, then it is
Trans.
trans. Okay. Naturally, this is more. This is more and healthier also. Trans is harmful. People who consume saturated fats and trans unsaturated fats, they get heart diseases easily. Understood?
Yes.
Yes, sir.
Right, for simple example, now at my place, in my home, I have instructed my wife, if you fry some food in oil, after frying first time, don't use that oil for a second time frying. Don't use that oil for, what is it? A second time frying. If you use it for a second time frying, in the second time, that this unsaturated fat has turned to trans unsaturated fat. So what are the harmful ones among the fats for the heart?
Trans.
Saturated.
One, it is saturated fats are harmful. One, it is, what is it? Saturated fats are harmful. Second,
Trans
trans
unsaturated.
unsaturated fats are harmful. So the only harmless fat part is
Cis unsaturated.
this unsaturated fat. Clear? Are you clear?
Yes.
Diagrammatically, they will not ask you anything. Don't need to take these diagrams and those things. Diagrammatically, they will not ask anything. Okay. Here, I have given you a note. Fatty acids are there. There's a carboxyl group. There's a hydrocarbon part there. In the hydrocarbon part, 16 to 18 carbon atoms will be there. Hydrocarbon is the one responsible for the hydrophobic nature of the fatty acid, and it is water insoluble. According to the presence and absence of double bonds in the hydrocarbon chain, we classify fatty acids as saturated fatty acids and unsaturated fatty acids. If you take unsaturated fatty acids in the hydrocarbon chain between the carbon atoms, there will be one no more double bonds will be there. In those places, the double bond available places will be actually bending, and those bent places are called by the name kinks, K-I-N-K, kinks. Unsaturated will be kinking because of the double bond, but saturated ones will not be kinking, they will be straight molecules. Okay, unsaturated fatty acids are present in olive oil in among the plant oils and cod liver oil among the animal oils. Saturated ones are having only single bonds there. Okay, in this place, in this story, okay, we have not included this story, but you have to now include it. Okay, unsaturated fatty acids are two types. Unsaturated fatty acids are two types. What are the two types of unsaturated fatty acids? One, cis
Yes.
unsaturated fatty acid. It is beneficial. It's beneficial. And second, what is it? Trans unsaturated fatty acids. They are harmful. Clear?
Clear, sir.
You can mark it down. Have you marked it?
Yes, sir.
Sir.
So let's mark another point there. Okay. Saturated fat and trans unsaturated fat, okay, bring, okay, or contribute to atherosclerosis. Saturated fat and trans unsaturated fat contribute to, what is it? Atherosclerosis. Have you marked it?
Yes, sir.
Now we are coming to the triglyceride formation reaction. The triglyceride formation reaction. Okay. In the triglyceride formation reaction, I will show you a triglyceride formation reaction. This is okay, but it has been given okay, but okay. There is one in the resource book. Okay. Better you children practice each and every diagram in the resource book. This is the triglyceride formation reaction. Okay. Here on the left hand side, what it is given to you is the glycerol molecule. Okay. On the right hand side, it is given to you. How many fatty acids molecules? How many fatty acid molecules?
3.
3. R is representing the hydrocarbon chain. Okay. R is representing the, what is it? Hydrocarbon chain. Here you can see they are reacting with H of the glycerol and OH of the fatty acid to form water molecule. How many water molecules will be formed in this manner?
3.
3. So we have to write in this place how much? 3. After marking it, this is the bond getting created, and that is the bond called, which bond it is? Which bond it is?
Ester bond.
Ester bond. Okay. So you better in your white sheet, you better mark this one down. In exam paper, they will directly ask you, write this total structure, how the acyl glycerol molecule is formed. Okay, go ahead and tell me after completing. Tell me after completing.
Done, sir.
Good. So, actually, that is the resource book 1. So this is looking very neatly giving ideas, but syllabus no. Sri Lankan syllabus mean we have to stick to those ideas. Thereafter, you are coming to the heading called home,
Phospholipid.
phospholipid. In the case of phospholipid, this is the diagram that represents the phospholipid. In this diagram, let me introduce you one by one. Right here, in this place, there will be a glycerol molecule. It is the glycerol molecule. To the glycerol molecule, 2 fatty acids are bonded. How many fatty acids?
2 fatty acids.
2 fatty acids are bonded. To the third carbon, there will be a phosphate group connected. To the phosphate group, there will be a nitrogen containing choline group connected. According to your syllabus, that choline is not a compulsory group in all the phospholipid, but majority of the phospholipids will carry whom? Choline. So what are the molecules contributing to make a phospholipid molecule? Just tell me the list.
Choline, phosphate,
Choline.
glycerol,
Choline, then
phosphate,
phosphate,
Glycerol.
glycerol,
2 fatty acids.
and fatty acid. Okay, how many types of molecules are contributing? How many types of molecules are contributing?
4 types.
4 types. How many fatty acids are contributing?
2.
Fatty acids.
2. Okay, if you look at the phosphate group, can you notice a minus there?
Yes, sir.
Yes. If you look at the choline, Can you notice a plus there?
Yes, sir.
Yes. Having plus minus is representing, it is polar. Understood?
Yes.
So, what are the polar regions in the phospholipid?
Choline and phosphate.
Choline and phosphate. Clear? Choline and phosphate are polar portions in the phospholipid. Okay, and right. So, if you take this whole, they are dividing the total phospholipid into two major regions. What are the two major regions? What are
Hydrophilic
the
head.
two major regions?
Hydrophobic.
Hydrophilic head, hydrophobic tails. How many hydrophobic tails are there?
2.
Two. Who are the one contributing to form the hydrophobic tail?
Fatty acid.
Fatty acids, hydrocarbons, correct? Who are the ones contributing to make the hydrophobic head?
Choline, phosphate, glycerol.
Glycerol, choline, phosphate, and what is it? Glycerol. Hydrophobic head, it's a polar part. Hydrophobic head is a, what is it? Polar portion. Hydrophobic tails are, what is it?
Non-polar.
Non-polar. Okay, and remember, phosphate group is, what is it?
Acidic.
Acidic. Phosphate group is acidic. Right? Keeping this one, various kind of questions can be given to you. Anyway, one promise is that they will not ask you to draw this structure. Understood? Have you marked these details?
Yes.
Right. So, what are the elements making phospholipid? What are the elements making phospholipid? Look at the diagram and you can tell what are the elements making phospholipid.
Hydrophilic head and hydrophobic tail.
Elements now, hydrogen, helium, lithium, beryllium, boron, carbon. They are the one called elements.
Carbon,
What are
hydrogen,
the
and oxygen.
carbon? Look at the diagram. Carbon,
Carbon, hydrogen, oxygen, nitrogen, phosphorus.
phosphorus. What is the group responsible for the nitrogen? What is the group responsible to give the nitrogen element?
Choline group.
Choline group. If choline group is there, only nitrogen can be expected. Correct?
Yes, sir.
So those ideas you have to understand. Right. So please go through this note and see whether is it clear. Clear?
Yes, sir.
Now coming to the functions of lipids. One function, food reserve and energy source. Who are the ones working as food reserves and energy source? Who are the ones working as the food reserve and energy source?
Triglycerides.
Triglycerides or fats and oils. Second one, maintaining the fluidity of cell membrane. Who does that work?
Phospholipid.
Phospholipid. Okay, put a comma and write home.
Cholesterol.
Cholesterol. Third one, acting as a signaling molecule, hormone. Okay. And fourth one, found as a component in, as a word to include that. Found as a component in animal cell membrane. Which cell membrane? Which cell membrane?
Animal cell membrane.
Animal cell membrane. Cholesterol is not noticed in home
Plant cell membrane.
plants. Okay, you don't see cholesterol found in plants. Okay.
Okay, sir.
Right. So with that one, lipid portion is over. Finishing the lipids, we are coming to home.
Protein.
Protein. Right. So regarding proteins, comparatively, we have a good idea. If you take proteins, what are the elements making the protein? What are the elements making the proteins?
Carbon.
Loudly. வாய்க்குள்ள பேச வேணாம், ஆனா கஷ்டம் எனக்கு. Why other people are not speaking?
Carbon, hydrogen,
Ah.
oxygen, nitrogen, and sometimes sulfur.
வாய்க்குள்ள கொஞ்சம் போவானாம். ஓகே, அட்வான்ஸ் லெவல்ல ஒரு சயின்ஸ் படிக்கிற புள்ள வாய்க்குள்ள கொஞ்சினா அதை விட ஒரு மோசமான நிலைமை யாருக்கும் கிடையாது. Right. So proteins are complex organic molecules. They are another set of biopolymers. They are polymers of amino acids. The characteristic bond that specializes them is which bond?
Peptide bonds.
Peptide bonds. Okay, nearly 20 amino acids are involving in making proteins. Every protein has a specific amino acid sequence. Okay, the sequence of the amino acids decide the biological function of the protein. If the sequence is altered, though the same amino acids are present, the protein's biological function will change. Okay, so please go through these points regarding protein. Okay.
Okay, sir.
Right. Here we speak about how does the peptide bond gets created. In the peptide bond formation, two groups involved. Okay, in an amino acid, there will be 5 separate portions we can identify. Here, this is the carboxyl group. This is the amino group. They are the one involving in making the peptide bond. Okay. Carboxyl group donates an OH. Amino group donates an H. And together they will form a water molecule. Together they will form a, what is it? Water molecule. Then between carbon and nitrogen, the peptide bond will be created. The peptide bond is created between the whom and whom. Whom and whom.
Carbon and nitrogen.
Carbon and nitrogen. Okay, it's a characteristic bond for protein. Clear?
Yes, sir.
Right. Anyway, you have to practice these diagrams. ஒண்ணு விலையும் கொள்ளணும் நீங்க. This is the right time. இதுதான் உங்களுக்கான the most potential period. Right? O level இருக்கிற அந்த எல்லா படிச்ச அந்த O level இருக்கக்கூடிய அந்த burden எல்லாம் ஒரு ஓரமா வச்சிட்டு மைண்ட் வந்து ஃப்ரெஷ்ஷா இதை பார்க்குற ஒரு மைண்ட் செட்ல உள்ளது இதுதான் ஸ்டேஜ். இந்த ஸ்டேஜ்ல நீங்க பெஸ்டா உங்களை ப்ரிப்பேர் பண்ணிக்கொள்ளணும். எந்த நேரமும் அந்த ஒரு மைண்ட்லயே தான் நீங்க இருக்கணும். உங்களுக்கு அப்படி வராட்டியும் அதை நீங்க உங்களுக்குள்ள சொல்லி சொல்லி கொண்டு வர ட்ரை பண்ணணும். அந்த மைண்ட் அப்படி நீங்க சின்ன பிள்ளைகள் தானே, ரைட்? என்னுடைய பார்வையில் சின்ன பிள்ளைகள் தான், ரைட்? குழந்தைகள் தான். அந்த ஒரு மைண்ட் செட் எங்களுக்கு இருக்கும், ரைட்? அப்போ விளையாட்டுத்தனம் உண்டு இருக்கத்தான் செய்யும். இப்ப நாங்க வந்து போற கோல் வந்து பெருசு. அந்த கோலுக்கான ரிவார்ட்ஸும் பெருசு. அந்த கோலும் பெருசு, அந்த கோல் உங்களுக்கு தார ரிவார்ட் வந்து அதை விட ரொம்ப பெருசு. ஸோ ஈச் அண்ட் எவ்ரி மினிட் நம்ம ஏதாவது பண்ணிக்கிட்டே இருக்க வேண்டிய ஒரு சிச்சுவேஷன் இருக்கு. ஓகே, அதை ட்ரை பண்ணிக்கொண்டே இருங்க. ஒரு டயக்ராம ட்ரா பண்ண, ஒரு டயக்ராம திரும்பி ட்ரா பண்ணி பாருங்க. ஏதாவது உண்டு அதோட இன்ட்ராக்ட் பண்ணிக்கொண்டே இருங்க, ரைட்? இதுதான் அதுக்கான சரியான நேரம். இன்னொரு 6 மாசமோ ஒரு வருஷமோ கடந்து நீங்க வந்து இதெல்லாம் செய்ய ட்ரை பண்ணீங்கன்றால் உங்களுக்கு நேரம் இருக்காது. எல்லா பக்கமும் வேலைகள் வந்து சூழ்ந்து நின்றுடும். அப்ப வேலைகள் டைம் மிக்காது. ஸோ இப்போதைக்கு கிடைக்கிற நேரத்தை இப்பதானே தொடங்கி இருக்கிறோம், ரைட்? இன்னும் ஓஎல் ரிசல்ட்ஸ் கூட வர இல்லை அப்படின்ற ஒரு மைண்ட் செட் உங்களுக்கும் இருக்கும். ஸோ அந்த மைண்ட் செட்டை பிரேக் பண்ணி கொண்டு நாங்கள் ஒரு கோலுக்காக இறங்கி இருக்கிறோம். அந்த கோல் நாங்கள் வந்து அச்சீவ் பண்ணணும். அது ஒரு பெரிய கோல், அது ஒரு பெரிய அச்சீவ்மெண்ட். அந்த அச்சீவ்மெண்ட்டுக்கு பிறகு உங்களோட வாழ்க்கை இன்னொரு தளத்துக்கு போயிடும். அதை நீங்க அச்சீவ் பண்ணிட்டீங்க என்றால் உங்களோட லைஃப் வந்து இன்னொரு ஃபிளட்டுக்கு மூவ் ஆயிடும். அதுக்கு பிறகு நீங்க சந்திக்கிற மனிதர்கள், அதுக்கு பிறகு நீங்க சந்திக்கிற சமூகம், அதுக்கு பிறகு உங்களுக்கான வாழ்க்கை, இது எல்லாமே புதுசு. இதே மனிதர்கள், இதே வட்டம், இது இருக்காது. அதுக்கு பிறகு உங்களோட உலகம் வேறு. இப்ப மௌத்துக்கு பிறகு இன்னொரு உலகம் வர மாதிரி வாழ்க்கையில இன்னொரு உலகம் தான் உங்களோட அச்சீவ்மெண்ட். ஸோ அதை நோக்கி நீங்க ட்ராவல் பண்ணும்போது முக்கியமாக அந்த டெடிகேஷனை வச்சு கொள்ளணும். அது ரொம்ப ரொம்ப முக்கியம். நீங்க வேலைலாம் செய்றீங்க, நான் வந்து குறை சொல்றதுக்காக சொல்ல இல்லை மகள், அப்படி எடுத்துக்கொள்ள வேணாம். டோன்ட் டேக் ஓகே, அந்த மாதிரி ஒரு மைண்ட் செட்ல சொல்ல இல்லை, ரைட்? நீங்க ஒவ்வொரு டயக்ராமும் ட்ரா பண்ணி பாருங்க, ஒவ்வொரு விஷயமும் செய்து பாருங்க. கடைசியில நேரத்தை மேக்சிமம் யூஸ் பண்ணிக்கொள்ளும். ஓகே, right. So what is our next heading?
Levels of protein structure.
Levels of protein structures. Okay, there are 4 levels in the protein structure. All are polypeptide chains. Protein means a polypeptide chain. Protein is a polypeptide chain. This polypeptide chain gets arranged in various ways. Okay, so what is it? Protein is a, what is it? Polypeptide chain. Many amino acids joined by many peptide bonds, a polypeptide chain. Okay, this chain undergoes several modification to form the levels of protein. Understood? Understood?
Yes, sir.
Protein is a polypeptide chain. That chain, it is undergoing several modifications. The modifications output is one level. If you see no any modification ondum illa, then it is a primary structure. Understood?
Yes, sir.
That means it is the only level without any modification. Just the amino acid chain is there. One end carboxyl group is there, other end amino group is there, just the chain there. That is which structure?
Primary structure.
Primary structure. Could you please write down this? Okay. Okay. You wrote it.
Yes, sir.
Right. In the secondary structure, there are modifications. According to the modification, they are dividing it into two types. See these two words. One they call alpha helix. Other one they call beta pleated sheet. What are the two forms? What are the two secondary structures?
Alpha helix, beta pleated
Alpha
sheet.
helix. Other one?
Beta pleated sheet.
Beta pleated sheets. Okay. If you take the alpha helix, you have seen, even you have climbed up a spiral staircase. Do you know a spiral staircase? Do you know a spiral staircase?
Yes, sir.
Yes. So if you take a spiral staircase, take this one. Here, it is going, here there will be an imagine, okay, here there is a central axis. Okay, it is moving around, what is it? That central axis. This is the spiral staircase. Like this, one chain will be spirally arranged, and that is the arrangement called alpha helix. Okay, so it is like a spiral staircase. If you go for beta pleated, what is it? Sheet. Okay, this is the arrangement of beta pleated. Okay, beta pleated sheet. Look at this diagram. If you take this beta pleated sheet, look at the diagram and tell me, is it a single chain or there are two chains there? Is it a single chain or there are two chains there?
Two chains.
Look at the diagram. Can you
Single
see
chain.
the screen? Can you see the screen? I have shared
Yes,
a,
sir.
yes. So in the diagram, there's a chain marked you, correct? Look at that chain. Is it one chain or there are separate two chains there?
One chain.
One chain. But the way it is arranged, okay, it is giving it a two chain that are parallel to each other. That means if you have closed this, now a person seeing it, what he will think? If you close in this manner, he will think there are two chains there that are parallel to each other. But in reality, it is not two chain, it is a single chain. Now you can understand here. Can you see there are two chains shown to you on the left hand side? Is that mean beta pleated sheet is having a two separate chains?
No.
No, it's a single chain arranged in that manner. Here, to keep this structure, to maintain this structure, there will be hydrogen bonds. Which bonds are there? Which bonds are there?
Hydrogen bond.
Hydrogen bonds are there. The purple color points are given. They are hydrogen bonds. Hydrogen bonds are created here between hydrogen and oxygen. Hydrogen attached to whom? Hydrogen attached to whom involved in making the hydrogen bond?
Amino group.
Check it. Hydrogen attached to whom involved in making the hydrogen bond?
Amino
Yeah.
acid.
Loudly.
Amino group.
Amino group, nitrogen. Correct? Oxygen belongs to whom? Attached to whom?
Carboxyl group.
Carbon. Oxygen, that is syllabus to you. Oxygen atoms attached to whom? Carbon. And nitrogen atoms attached to whom? Nitrogen, I mean hydrogen. Atoms attached to whom?
Nitrogen.
Nitrogen involved in making the hydrogen bond. So if you take the secondary structure, in the secondary structure, there will be hydrogen bonds and peptide bonds. Both bonds will be there. Clear? Clear?
Clear, sir.
Good. So who is the example given to you for alpha helix?
Keratin.
Keratin. Who is the example given to you for beta pleated sheet?
Spider silk.
Spider silk. That beta is compulsory. That beta is compulsory. That alpha is compulsory. Understood?
Yes.
Okay. Without alpha, without beta, you will not get score there. Okay. So that is secondary structure. Remember, in the amino acids, I think I will come to that one hereafter. Okay, the heading called amino acid there. Okay, in the amino acid, look at this diagram. If you take this amino acid component, in this amino acid component, this part is called backbone. So who are the components coming in the backbone? Mark the backbone. Mark the backbone. Have you marked the backbone? Have you marked the backbone?
Yes, sir.
Yes. The remaining part is called by the name side chain. So what are the two parts of amino acid? What are the two
Backbone and
parts of
side.
amino acid? Backbone and
side.
you might think suddenly why I am telling you this. Reason, in making that hydrogen bond, here we say the hydrogen bond is made now. Okay. The hydrogen bond is made with the contribution of the backbone. Clear? The hydrogen bond is made with the contribution of the backbone. Nitrogen in the backbone, carbon in the backbone, oxygen in the backbone, hydrogen in the backbone. Backbone makes the hydrogen bond. Clear?
Yes, sir.
Good. Let's we continue. I will send another short question set to you. Do and send it to me. Okay. When doubts arise, ask me. Go ahead in practicing it. Bye. Assalamu alaikum.